only mode.
Hello everyone and welcome to the QIAGEN's 4-part liquid biopsy webinar series on circulating cell-free DNA about the purification, sequencing, and the data interpretation. My name is Abhishek and I'm your host for today's webinar. So before we start the presentation, let's do some usual checks to make sure that you can hear us. On the right-hand side of your GoToWebinar toolbox, there is a box called questions, and if you could just type something there like hi or hello, so that we are sure that you can hear us properly. All right, that's great. I can see a lot of highs and hellos coming in, and I can hear you. So that's perfect. Then I think we are all ready to start the presentation. We will keep it in a listen-only mode just to make sure that there are no echoes. And if you have any question during the presentation, please feel free to submit them in the question box, and I will ask them to the speaker at the end of the webinar session. So with that, let me introduce you. Today I have Dr. Thornton Foster, who is the senior scientist in the PreAnalytiX R&D group, who will talk about the new technology and workflows for integrated collection, stabilization, and purification of circulating cell-free DNA. Dr. Foster joined the QIAGEN in 2004, and PreAnalytiX, as some of you might not know, it's a joint venture of QIAGEN and Becton Dickinson. He and his team are developing sample stabilization solutions and dedicated sample purification methods for human specimens. Thorsten studied at the Westfälische Wilhelms University of Münster, where he obtained her PhD in biology and Institute for Molecular Biology and Biotechnology of Fungi. So I think it's a great opportunity to have him here, and please feel free to ask as many questions you have during the session. If you're not able to answer them at the end, we will definitely answer you with an email. And now I will hand it over to Dr. Thornton Foster. Thank you for joining us today.
Okay, thank you, Abhishek. Thank you all of you and listening to my talk here. Yes, so we will have this slides now for the new technology we developed here at PreAnalytiX R&D for stabilization, collection, and purification of cell-free nucleic acid DNA from plasma from human blood. And I will show you what this means. So this is not only the technology, but also what this has consequences for the collection and also for the purification. So in the overview slide that is shown here, I give you a very short introduction in the field and why we need stabilization of ccfDNA in plasma and blood. And then I show you the main features of our new tube, where we integrated a very new stabilization technology made here in the PreAnalytiX R&D. And in the next part of a few slides, I show you then the integration of this tube and the plasma that comes from this tube to an isolation procedure on the state-of-the-art QIAsymphony robot that we have here. And finally, I summarize all of this in a few slides. So starting with the introduction, I choose this nice picture from the Nature Reviews article from Schwarzenbach et al. That shows you what is ccfDNA in blood all about, and I think nearly all of you know this. But I just want to highlight here some special features of the ccfDNA from body fluid that is important for the stabilization features that I present you in the slides in a minute, but also for the isolation with the QIAsymphony in the last slide. So as you all know, this DNA that you can recover from the blood and the plasma is strongly fragmented, and the most important part of it much less than 500 base pairs long, but what makes it a little bit more complicated for us as the developer of the isolation procedures. There are also large fragments or larger fragments, larger than 500 base pairs in this original ccfDNA, coming, for example, from necrotic processes or from organ diseases or cancer cells. And these are also of interest for many applications here. So we have a large spectrum with respect to length, and additionally that we have a very, very low concentration of this free circulating nucleic acid in body fluids, plasma, serum, urine, and others, far below 100 nanogram per milliliter for most cases. And we concentrate now this talk on the ccfDNA from plasma. In the next slide, I try to show you and to explain it to you why we have the need for stabilization of ccfDNA, especially in plasma, and what is the problem. So what you see in the graph and on this slide is a picture from an Agilent bioanalyzer with alleles of ccfDNA generated from plasma of an EDTA tube directly after blood collection. So the blood was collected in a normal EDTA tube, plasma was separated, and ccfDNA was isolated. And what you see here, there's a small peak here in just the front end of the graph, and this represents the normal ccfDNA. Most fragments are in this range of 66 base pair, but you see also a little smaller peak with a larger length of higher molecular weight. So this is the picture if you isolate directly your CCF from the plasma after blood collection, and what happens if you, for example, have to store the blood or to ship the blood or other things that can happen. This is shown here in this light blue graph here from EDTA blood from the same donor, stored for 10 days at room temperature, and then the plasma was separated from the blood cells, and ccfDNA is isolated. And what you see here is that you have a huge signal from ccfDNA coming from apoptosis of white blood cells. And the consequence of this is that your small amount of original ccfDNA is diluted by this huge amount of other DNA coming from dying cells. And this is the reason why you need this stabilization feature that what I will present to you in the next slide. Yeah, as I said, so in the next few slides I will present you the technology and the main features of our new PAXgene Blood CCF DNA tube, which will be available this year. And to start with this, I have a short look here in the workflow options for tube format that are commonly used in the field of ccfDNA from blood. So what you see here on the top of this table, we have, of course, the EDTA blood. It has no stabilization functionality, and the commonly often used track tube, so the track cell-free DNA blood collection tube, and then our new tube called PAXgene CCF tube on the right. And I try to figure out the most important features and workflow features. And starting with the first line is the blood draw volume and the plasma yield you get from blood. Of course, as most of the applications that are done with ccfDNA are relating to very few copies of DNA in the blood, you need, of course, a certain volume of blood. So 10 ml is what you get normally from EDTA blood, and 10 ml blood draw is also realized with the Strack tube and with our tube as well, resulting for all these tubes in a plasma volume of at least 4 milliliters. The next slide is about the tube material. So the tube material is very important, especially if you think about transportation and breakage of tubes. And for the standard EDTA tube that are in the field, you have normally a plastic tube. The STRACT tube is made of glass, which is, can cause some issues, especially during transport, cracking of the tube, but also we got reports from the field that some of the tubes also break during centrifugation. So this is the reason why we decided to make our tube from plastic. So this is comparable then to a standard EDTA tube from the material side. So coming from these overall features now to workflow features that are more tightly linked to the ccfDNA. So as I already mentioned, the EDTA blood contains no stabilization reagent in it, and therefore you have to separate the plasma from the blood cells as fast as possible. So this could mean, of course, that you have to directly isolate the plasma from the cells in the doctor's office, or you have to guarantee a very tightly monitored cooling chain. But even then, you have only a very restricted time to transport the blood. This is not the case for the STRACT tube. There you have a stabilization solution in it, which stabilizes the white blood cell and therefore prevents apoptosis of these cells. And we, for our new tube, also have a stabilization solution in the tube that have the same features with respect to white blood cell apoptosis. So then, of course, very closely linked to this centrifugation point is the transport. So as I mentioned already, there's no room temperature transport for EDTA possible for most applications, but this is of course possible with the STRACT tube and with the new tube as well. So regarding the stabilization solution that is in the STRACT tube and in our tube, we avoided to use any cross-linking reagents in our stabilization solution, so that you have no impact on the ccfDNA with respect to your downstream. And what is the next on this row, on this table, is a standardized workflow. So this is very important because you have to think that very minor things that you do different to other labs can have a strong impact on the yield and also on the population of the ccfDNA you can isolate from the plasma. And therefore we think that standardized complete workflow, which means we have a tube with a dedicated ccfDNA isolation procedure dedicated for this tube, and you have everything from one supplier as the best solution. And this is what we wanted to, what I want to present you in the later slides here. And very closely linked to this, if you think in higher throughput of samples, the question for automation and an automation that is compatible with most of the clinical workflows as well is mandatory. And therefore we linked our new tube with the stabilization solution closely to our QIAsymphony platform and developed a dedicated protocols and kits for this tube, so that you have a streamlined workflow starting from the collection over transportation, plasma generation, and PCF isolation in the end. Yeah, so coming from this overall features to the data that I want to show you and that I want to show you to convince you that we developed here a really good stabilization solution for your needs. So I start to show some data here which shows that we prevent even all, besides the white blood cells, also red blood cells from lysis, so we prevent hemolysis. So this is shown in this graph here. So what we have done, we isolated the plasma from our tubes and also from EDTA tubes drawn side by side after different time points of room temperature storage. And what you clearly see here, this is the absorption of about 414 nanometers. This is the maximum absorption of hemoglobin. And you see that with the EDTA blood plasma you see here in blue, you can get an increase over time of the hemoglobin this therefore for hemolysis, while the plasma from the PAXgene tube stays more or less the same with respect to hemolysis, and so therefore we prevent hemolysis. Okay, so hemolysis is of course a point where you can say, red blood cells have no DNA, and therefore why should I care about hemolysis? And this is shown in the next slide. Hemolysis can of course impact your plasma preparation. So this is an example picture that was sent to us from a collaboration partner, LifeCodex, and what you see here is blood drawn into a PAXgene blood collection tube on the right and a streak tube for this example on the left. And you see that this is very reddish, and this is of course, as you can imagine, can have an impact on the plasma you can remove from these tubes safely, and so that you really do not disturb the body code between the red blood and the blood cells and the plasma. So if you get such tubes, and this is sometimes the case for other tubes and for EDTA tubes, as I showed in the picture before very, very clearly, can have an impact on your plasma yield and therefore also have an impact on your ccfDNA yield after that. Okay, so coming from hemolysis more to the apoptosis of white blood cells. So white blood cells, of course, have to be prevented from apoptosis so that that the ccfDNA is not DNA yield is not diluted. And in the first experiment that is shown here, we looked at the point: is there really apoptosis, and how can we visualize this? And what we have done here is again collected blood in EDTA tubes and new PAXgene tubes and stored these tubes for up to 10 days, isolated from the whole blood the genomic DNA, and that is shown on the left directly after blood collection, and on the right after 10 days of storage. And if you do this, you see, okay, here for the EDTA blood, you see this apoptotic band, so clear evidence that you really have a strong apoptosis here, while for the PAXgene ccfDNA tube, you do not see these bands. Okay, so this is a picture from genomic DNA from whole blood, but we're dealing about ccfDNA from plasma. And in the next slide, I'm coming back to the picture I showed you in the beginning, and this is again the DNA isolated from EDTA tubes, from the plasma of EDTA tube from 0 directly after collection of the blood and after 10 days. And what happens if you draw in parallel blood of the same donor in the PAXgene tube? And this is shown here with the greenish lines. So clearly you see in this Agilent picture that we have no induction of apoptosis, not in the plasma. We see the same bands of the ccfDNA which were original in the EDTA blood here for the PAXgene tubes after 0 days, so directly after blood draw, as well as 10 days. So this is the Agilent picture, but we of course have done other analysis to further demonstrate this. And this is shown in this picture here. So we have done with the areas that were shown in the picture before, done PCR, and we developed an assay that is targeting for the 18S rDNA. And this is a duplex assay, so with one amplicon of 66 base pairs, so representing the short fragment, and an amplicon of 500 base pairs, representing more the larger fragment, and additionally the apoptotic fragment that's coming if you apoptosis in your blood. And on the left part of this figure here, you see what happens during EDTA, very similar to what is shown in the Agilent. You get a strong increase over time at room temperature storage for the different for the two different amplicons, which is shown here on this axis here. So the ratio between the day 0 copy numbers to the compared to the different time points that are shown here. You see that after 10 days you have 100 to 300 induction of fold change over time for the EDTA tube. And if you look at the same blood from the same donors, isolated plasma here with CCF isolated from this plasma, you see nearly no change over time up to 10 days. So again, a strong evidence and a clear picture that our new tube really prevents apoptosis from the white blood cells. Okay, so this is PCR, and of course I know that many of you use now NGS as a downstream, and therefore we started a collaboration with the company LifeCodec, very good partners of us, and started with them together a research study with maternal blood. So we collected 18 specimens of maternal blood, parallel the track tube and our tube were drawn one after the other in the doctor's office. The blood was shipped to LifeCodec, and they isolated then the plasma and the ccfDNA from this plasma, and then compared the two sample types, STRAC and PAXgene. And first of all, they looked in their QC assay, which is again a PCR assay here on the left part of the three pictures in the bottom of the slide. And with this assay, they can look for the fetal fraction of the ccf DNA. And this is done by a methylation-based assay. I cannot go into details here, but their quality criterion is that the CCF fraction, that the CCF DNA have to be at least of 4% of the fetal fraction, so 4% of the minimum of 4% of the CCF to be CCF DNA have to come from the fetus. And what you see in the left picture here on the bottom of the slide, that we have absolutely comparable amount of fetal fraction with our new tube shown here in green and the STRAC tube in gray. So we are far beyond this criterion of 4%, and the fetal fraction is comparable between these two systems. Then we, from these LOS libraries were prepared, and they were fueled into the NGS workflow at LifeCodec. And these are the two pictures on the right here. So first, in the middle, we see the total reads that could be generated from these two different sample types, and you see that this is absolutely comparable. As also comparable is on the right the amount, the portion of mapped reads and the portion of unique mapped reads as shown in this picture here. So overall, absolutely comparable data for the QC and the NGS workflow, which also comes out if we look at the results from the from these NGS data. So the fetal gender was concordant between collection tube spore specimens for both samples we had, and also if we look at all 18 specimens that were collected, the NGS results of the prenatal test from LifeCodec were absolutely concordant with the current method and were all negative for the 3 trisomies that were looked at. Okay, coming back to the tube itself and to the things that make it different from other tubes in the field. We have seen, and I showed this in the previous slide, that the results from these tubes are very comparable to other methods. But what is the difference now between this tube and what is this all about? So you see, I put here 4 things, and now I show you what is the most important difference here. So we, as I said, we use the plastic material that avoids the tube breakage during transport or centrifugation. So this is a safety feature. Also, a safety feature here is the safety VD hemoguard closure. So the closure here on the top of the tube is not just a rubber stopper here, but also this plastic shield. So if you open the tube, this prevents the blood from spreading around and therefore helps you to avoid contamination with blood your lab coat or your table, and of course to other tubes that stand near to this tube. The third point is the prevention of hemolysis. As I mentioned already, this helps you to get the maximum yield from plasma separation from a tube, so you can really clearly distinguish between the plasma phase and the buffy coat phase and have no issues with this. And the fourth point, as I already mentioned, we, for our stabilization solution, have no cross-linking agents or any other agents that modify or can potentially modify your ccfDNA, and therefore we have the safety feature included that you have no issues with your sample preparation, and especially not for your assay performance. Okay, so the next part of my talk will show you the link from this tube that I explained you in the last slide to the dedicated QIAsymphony workflow, which will also be available in this year. And to put it in the right light and to explain it, what are the differences, for example, to other solutions that we have here at QIAGEN already, I will explain this slide to you. So what is shown here is the complete workflow from the patient to the clinical result if you use ccfDNA. And on the top here you see the two tubes that are mainly used at the moment in the field, so the EDTA tube and the STREC tube. And if you use these tubes, you can use it with the current QIAsymphony Circulating nucleic acid kit that is in the field already since about last year. And this kit and protocol is a generic protocol. So with this kit and protocol, you can use nearly all currently available plasma material and isolate ccfDNA very efficiently. So as I said, this is a generic approach. There is nothing really told you about plasma isolation, about transport, because this is just dealing with the ccfDNA isolation from the plasma that you get. Our approach is a little bit different. So we, as I said, we isolated, we generated a new tube with a new stabilization solution, which has some safety feature as mentioned. And with this tube, we address also the transport, as I said, so 7 days in the room temperature, no issue. Transport is in plastic tubes, so no breakage of the tube. And we also address the workflow of plasma centrifugation, plasma separation. So the centrifugation protocol was generated by us, and we have also addressed this. And finally, we have a dedicated isolation kit and protocols for the QIAsymphony machine, so that we have a system approach here that integrates the complete workflow from blood collection to the isolation of ccfDNA from plasma. So how is this done on the QIAsymphony? So we have two options for each protocol line that we will launch. So we have a protocol for a relatively low amount of plasma, 2.4 ml of plasma. If you choose this, your plasma will be pipetted on the QIAsymphony consumables, and you will add, of course, the proteinase K binding beads and binding buffer. And mixing this, you have a combined binding and proteinase digestion step in the beginning. And after that, you have bead separation, and then you have different wash steps, 3 wash steps to get rid of contaminants. And finally, you have an elution. And for our system, you have the option to choose between 3 different elution volumes. If you are interested in higher plasma volume, you have also the possibility that you start with 2.4 ml and choose the 4.8 ml plasma protocol, and then after the first liquid of 2.4 ml of plasma was processed, during this process here, the beads were separated, and then we used for a second aliquot of 2.4 ml of plasma, proteinase K was added again, binding buffer was added again, and then you have a second binding step here, and then separated beads go through these wash steps and find the elution. So we have two options, a high plasma and a low plasma volume protocol here. In the next slide, I just want to describe a little bit more in detail what distinguishes the QIAsymphony procedure of the PAXgene tube from the generic protocol as described 2 slides above. So as I said, we have a dedicated isolation technology that was focused on our new tube and the stabilization solution that is in the tube. This means that we have a dedicated binding chemistry here optimized for the new tube, and we also have some steps optimized to facilitate this plasma generated from this tube. This means basically the binding steps and the elution steps. Furthermore, we, in contrast to the generic kit, we have 2 protocol lines. So as I mentioned in the very beginning, we have the situation that we have downstream application for customers who are concentrating on the small fragments of ccfDNA, but there are also applications in the field that using the large fragments, mainly from necrotic processes. Therefore, we decided to have 2 different protocol lines, all with 1 kit, but 2 different protocol lines. The first protocol line is a so-called standard protocol line, focusing mainly on the short fragments, and this is what also the generic kit does. So this is very comparable in the outcome to the generic QIAsymphony circulating nucleic acid kit. But we address in the second protocol line also the large fragments. So therefore we call it logically the large fragment protocols, and they enabled, through a modified binding chemistry, a very efficient co-isolation of large fragments. So you get the same sensitivity for your small fragment, but additionally get the large fragment in your analysis as well. And finally, what is also different to the generic protocol, we offer 3 different elution volumes, 60, 100, and 150 microliters in the standard protocol. So finally, we have 2 protocol lines with 2 protocols in each line with a large volume and a small volume of plasma. So here is an example for the performance of the QIAsymphony CCF isolation procedure. So on the left part of this figure, you see the isolation of PAXgene plasma with the new PAXgene QIAsymphony kit, and on the right side you see the isolation of the generic kit using PAXgene plasma as well. And what you see there's a very similar performance, but you also might see that for all donors we have a slight advantage for the PAXgene QIAsymphony kit, and this is what I meant. So we have a dedicated solution. This brings a little advantage, not a very large difference, but you see a slight advantage with the dedicated kit. Okay, so finally, I like to summarize what we heard in my talk here. So overall, we have now for QIAGEN and PreAnalytiX a sample-to-end site CCF workflow for blood, which addresses all the needs that we think customers have with ccfDNA for plasma. So what you see, we have the new tube, as I mentioned, and this tube can, the plasma from this kit be isolated and then used either with the manual QIAarm circulating kit. So if your load or your throughput is not so high, you can use this QIAarm normal manual kit. But if you have a higher throughput and you like this idea about a closed system and an integrated system, you can use the plasma from our new tubes with the new QIAsymphony PAXgene Blood ccfDNA kit to get ccfDNA. But even if you think, or if you need for certain application EDTA plasma or plasma from extract tubes, this will be addressed via the generic QIAsymphony circulating nucleic acid kit. So overall, with this solution, we think that we have a complete pre-analytic workflow addressed by QIAGEN and PreAnalytiX. Okay, so this was my final slide. I thank you for your attention, and I'm happy to answer your questions.
Thank you, Thorsten. I think that was very informative, and I can see people have a few questions, and I think I can go one by one. Some of the questions I will try to compile them, because a lot of people were interested in knowing that how they can get the access to the protocols that you have mentioned, and if they are on QIAGEN.com.
Yeah, so this protocol, and so this the focus was on the technology presentation here. So as I mentioned, our plan is to have this available this year, but I cannot give a concrete launch date for this because from R&D I can say that the tube will be a little earlier, so that you can, or that the customers can validate and look in it and compare to existing workflows. And this will be in the first half of this year, and in the second, perhaps later second half of the year, we will also launch the complete system with the QIAsymphony kit and the protocol.
Okay, thank you. I think that also answers some of the other questions where people were asking about the release date of these tubes and kits. So I think you already answered that. So yeah, please keep keep a look at the emails from QIAGEN or on the QIAGEN.com, and you will see some announcements too.
Yeah, this is of course also announced on the homepage I mentioned here. So PreAnalytiX own homepage. There will also, of course, news like the launch and release address.
Absolutely. Thank you. Thank you for adding, Thorsten. The next question is about when you showed the data in the beginning of about the apoptosis of the white blood cells. The question is, what about the tubes like PPT with gel from BD? Have you also evaluated them?
Yeah, so we have looked in this, but the issue with this is that this gel, sometimes some blood cells attached to this gel, and then can be released in the plasma. Yeah, after the cell barrier is formed, and this can of course need to travel, and therefore we have not included this here.
Okay, thank you. The next question that I have on the table is, which a couple of people were interested in, that can they use the frozen plasma with the QIAsymphony circulating nucleic acid kit for isolation?
Yeah. This is a very good question, and the answer is yes. So you can of course separate the plasma and freeze it, and after thawing it can be used in nucleic and kitted, of course.
Okay, great, thank you. The next question is, can you also isolate germline white blood cells DNA using the PAXgene tube? I'm not sure if got the question.
This is really a tough question. We never looked at this. Sorry.
Okay, no problem. Thank you. I'm just going through the other questions. Some people want to know. Yeah, another interesting question is, does these tubes work with the urine samples as well? It's a strange question.
No, so these tubes are only for blood and plasma. No, so this is something that we perhaps will address in the future, but at the moment this is only for blood.
Okay, thank you, Thorsten. The next question is actually a bit specific on the kit. I mean, I will read it out. Maybe you can answer, or maybe we can reply back. So, what exactly is the difference between the circulating nucleic acid kit? I think they're talking about the QIAM versus the PAXgene blood ccfDNA kit. If both are from blood collected. If both can be used with the blood collected from PAXgene blood ccfDNA.
Yeah, so this is a good question. So the manual kit, so the QIAGEN kit, is based on a cartropic alcohol chemistry. So silica material and binding via alcohol and cartropic. And the QIAsymphony application is completely different. So it's an anion exchange matrix, and therefore you have no alcohols and no cartropic salts in it. From the binding chemistry and the binding matrix are completely different systems.
Okay, thank you. And I think for the details, people can always go and look at the resources on the website for both the products. The next question that I have is, like, how long can you store actually blood in these tubes if, let's say, at the room temperature or at 4 degrees Celsius or frozen?
Yeah, so the blood can be stored and shipped up to 7 days at room temperature. We have not launched. We are just in the mode that we also in different other temperatures, but this I cannot really say at the moment. But when we launch or when we see this, of course, we will have the answer for this. One of the claims that we made. What about other temperatures? But 7 days at room temperature, the blood can be stored in these tubes.
Okay, thank you. And a lot of people want to know how can they get access to the presentation. So what we will do, we will send around a link in few days, and then you can download the PDF document, and you can always get back to us after reviewing it if you have any more questions. The next question is probably it might not be for you, but people just want to know that how they can compare the price as compared to the EDTA or STRAC tubes.
Yeah, sorry, that I cannot really answer.
No, that's
so
fine.
I'm from R&D and sorry.
No, but yeah, I mean, people, I would say that just keep an eye on when these are launched, or you can then you can always get in touch with your sales reps in your region or send an inquiry to on the website. Well, with that, I think we are towards the end of the session. I think it was very informative and very good discussion, the kind of questions and the interest that people have for this. So I just want to remind everyone that we still have 3 more webinars coming in this part every week, which will take you to the next steps of isolation of cell-free DNA, followed by an NGS webinar and the data interpretation. So please keep tuned in, and we will also look for the other webinars from QIAGEN. And with that, thank you again, Dr. Thornton, for joining us today.
You're welcome.